Hopefully you aren't feeling too overwhelmed at this point. As I said, please use your study guide summary sheet for cellular respiration. This will help you keep track of things because we're really going to get into the nitty-gritty now on the rest of cellular respiration. The next two steps, the citric acid cycle, also known as the Krebs cycle, and oxidative phosphorylation, are where we finish the breakdown of glucose, have released all of the electrons, all of the energy that we're going to get out of it, and we're finally going to produce a whole bunch of ATP. So we finished up the last video with pyruvate oxidation. So you can see the product of that, the acetyl CoA, at the top of the image on the right there. Remember that glucose was broken into two pyruvate molecules. Each of those is converted into an acetyl CoA. So the citric acid cycle that we see here is actually going to spin twice for every glucose molecule. One glucose, two pyruvate, two acetyl CoA, two turns of the citric acid cycle. So when we talk about this, we're actually keeping in mind that this is spinning twice, and so all of our products are actually doubled. So the citric acid cycle is important because it completes the breakdown of glucose. So acetyl CoA enters the citric acid cycle, and ultimately, the last of the original carbons that were part of glucose are released as carbon dioxide. So we have now extracted all of the energy that we're going to get from that glucose, and we've released it as carbon dioxide. We can see there that we produce some NADH molecules and an FADH2, so that's our first experience with that guy. He's just another electron carrier. So those electron carriers are accumulating electrons, which means they're accumulating energy potential, and they are going to be used to produce ATP. You can see there at the bottom we also produce a little bit of ATP. This is ATP by substrate level phosphorylation. But the ATP is not the goal of the citric acid cycle. Those electron carriers and the final breakdown of glucose are really the goal of the citric acid cycle. So this is the complicated version of that nice cartoon picture that I just showed you. Again, you don't have to learn all of these molecules, but you do need to know a few key ones. So acetyl CoA comes in, it combines with a molecule called oxaloacetate. And when it does that, it produces citrate, which is where we get the name the citric acid cycle. So those are the two molecules that I need you to know for this. Acetyl CoA comes in, combines with oxaloacetate to produce citrate. In this process, electrons are removed, they're added to our electron shuttles, and bonds are rearranged through this cycle of carbon molecules. So these are all carbons that are not the original glucose that we had, but they are all part of this cycle, and they are moved around and shifted in order to remove electrons from them. So the goal of this cycle is to finish the breakdown of that acetyl CoA and ultimately regenerate that oxaloacetate so that a new acetyl CoA can come in. We can get electrons off of that. And so that's why it's a cycle. We just reproduce the oxaloacetate. So these are all the key components of the citric acid cycle. As I said, you need to know those three molecules there at the top. You don't need to know their structures. You just need to know that they combine and are part of the cycle. You can see here for every acetyl CoA that comes in, we get 3 NADH molecules and 1 FADH2 molecule, so 4 electrons are removed, and those are going to be used in oxidative phosphorylation. We also have carbon dioxides released. So for every acetyl CoA that comes in, we get 2 carbon dioxides released, and then we get a little bit of ATP produced. So we are trying to accumulate ATP. We just don't get a lot of it during this cycle. If you look at the bottom down there, we also can produce something called GTP. This is another molecule that's very similar to ATP that can be used in the cell as energy. More commonly, ATP is used, but GTP is produced in some organisms as well. So at this point, we've completely broken down glucose. So we're done with glucose now. All of the energy that was held in that glucose is now being stored in our electron carriers. So if you haven't written down your products for all of these, you may want to do that right now. Remember, I asked that you know inputs, outputs, and quantities. So our final step in cellular respiration is to finally make ATP, to finally remove the electrons from those carriers and get their energy out. Because the ultimate goal is to make usable energy for the cell, and that comes in the form of ATP. So here we have our overview of pyruvate oxidation and citric acid cycle. I have everything summarized for you. I'm not going to read this to you. You can write everything down as needed. So our last step is oxidative phosphorylation. So remember, oxidative phosphorylation is one of the mechanisms by which we produce ATP. There's a lot of new words on this slide. During oxidative phosphorylation, a process known as chemiosmosis uses electrons to run the process of ATP synthesis. So we're going to go through each one of these and talk about what is actually going on in this process. During this process, an electron transport chain, which is just a chain of molecules that gain and release electrons, pass those electrons and slowly release their energy. They are released from our electron shuttles, our NADH and FADH2, to an electron acceptor molecule. In general, this is going to be oxygen. This process occurs embedded in that mitochondrial membrane, that inner membrane, the cristae that we talked about. This process of oxidative phosphorylation is a series of proteins that are stuck inside that membrane. If you are a eukaryote, there are some prokaryotes that perform cellular respiration. And if you are one of those, you don't have a mitochondria because you don't have any organelles, but you do have a plasma membrane that surrounds the cell. And so the process of oxidative phosphorylation is going to occur embedded in the plasma membrane rather than the mitochondrial membrane. We're going to stick to the mitochondria though because we're eukaryotes and humans are quite self-centered, so we're going to talk about ourselves for a while. All right, so the electron transport chain is really the link between the citric acid cycle and oxidative phosphorylation. In the electron transport chain, we have a series of molecules that are embedded in that mitochondrial membrane. And each one of those molecules is more electronegative than the last. Remember, an electronegative atom or molecule is one that is going to pull electrons toward itself. And so as they are lined up next to each other, they're going to pass electrons to their neighbors because each neighbor is more electron hungry. Each neighbor is more electron greedy. And as those electrons are passed between molecules, their energy is slowly released. So NADH comes in. It's the least electronegative. It wants to give up its electrons, and it gives it up to that first protein complex there, the purple kind of bubbly shaped molecules. Those are big proteins that are going to take those electrons from NADH. You don't have to memorize all of those. You just have to know that each one is more electronegative, and so it pulls the electrons from its neighbor. So NADH deposits its electrons at the beginning of the electron transport chain. FADH2 deposits its electrons just a little farther down. But either way, they both dump off their electrons to molecules that want them, and then those molecules are passed down through this electron transport chain. What you're seeing here is actually a graph. It's an energy graph that shows the energy contained in those electrons. And we'll see how that energy is actually lost, where it goes in just a minute. But ultimately, those electrons are passed to oxygen. So here we finally see oxygen. We breathe in oxygen, and that oxygen is required for the process of cellular respiration. So we're finally going to use it. When those electrons are passed to oxygen, we produce water. So water is actually a byproduct, as is carbon dioxide, of cellular respiration. Remember, oxygen is really electronegative. It's the most electronegative atom, and that's why it's the final electron acceptor. When we say that an organism performs anaerobic cellular respiration, we are talking about an organism that uses something besides oxygen down here. It might be nitrogen, it might be sulfur. It's another electronegative atom that wants to take those electrons. So the process is the same, but some organisms use a different electronegative molecule down there to grab those to grab those electrons. So as those electrons are transferred into our electron transport chain, they lose energy. The reason that they lose energy is that energy is actually used to produce a hydrogen ion gradient. So remember, in chapter 5, we talked about active transport that requires energy, and one of the things we talked about was a hydrogen proton pump. And I said that the proton pump would be really important. The reason that it's important is as those electrons are passed through the electron transport chain embedded in the mitochondrial membrane, the energy from those electrons is used to pump hydrogen ions to the outside of the mitochondrial membrane. So remember, we have the mitochondrial matrix, which is the inside of the membrane. And so the hydrogen ions are pumped across that membrane, not outside of the mitochondria, but just outside of the matrix. And when that happens, you get something called chemiosmosis. So remember, osmosis is actually the movement of water. So in this case, we're moving hydrogen ions in chemiosmosis. It's a specific type of movement. And during chemiosmosis, we get the accumulation of those hydrogen ions. So what that does is accumulates a concentration gradient, but it also accumulates a bit of a charge gradient because those hydrogen ions are, because they're being moved by active transport, are accumulating. The concentration of those hydrogen ions is getting higher, but there's also a lot of positive charge that goes with those ions, and that's going to be used, that hydrogen ion gradient is going to be used to actually power the production of ATP. So as NADH drops off its electrons, some of that energy is used to pump hydrogen across. Electrons continue to be transferred. FADH2 transfers electrons, so we have more electrons moving through this transport chain and pumping their hydrogen ions across. Remember, ultimately our oxygen acquires those electrons and water is produced. Finally, we get to ATP synthesis. In this step, ATP is produced by something called oxidative phosphorylation. Oxidative meaning an oxidation reaction. The transfer of electrons is used to phosphorylate ATP. Remember our ATP cycle, where you can produce ATP by adding a phosphate to ADP. And so that's what we're going to do here. In the last slide, I showed you that hydrogen ions were accumulating. We were building up a concentration gradient. And if you remember that when you have an area of high concentration, whatever you have wants to move to an area of low concentration. And so we're going to use that concentration gradient to produce something called proton motive force. That means that protons are generating movement by their traveling along their concentration gradient. So we have two pictures of the molecule that does this. This is ATP synthase, and it is essentially a molecular turbine. It actually works like a rotor on, say, a wind turbine or a windmill, something like that. And the movement of those hydrogen ions along their concentration gradient is actually going to power the movement or the turning of that ATP synthase rotor. So it essentially acts like a motor that spins this molecule. It's really quite cool. So up here we have hydrogen ion concentration. Hydrogen ions funnel through that protein, and when it does, it causes the protein to spin, and the hydrogen ion moves down along its concentration gradient. When that happens, you actually get mechanical turning of the bottom, and when that protein turns, it phosphorylates ADP and produces ATP. This ATP synthase molecule is the protein that's doing all of the ATP production, and it is this oxidative phosphorylation step that uses chemiosmosis and proton motive force to produce the bulk of the ATP in our cell. Let me go back to that real quick. During this process of oxidative phosphorylation, we produce about 32 ATP from that gradient that is produced by the breakdown of glucose. So each glucose molecule can produce about 32 ATP in oxidative phosphorylation and about 4 in substrate level phosphorylation. So we get a total of roughly 36 ATP for a single glucose molecule. So as I said, we also have this product of this process of anaerobic respiration. It is the same process: glycolysis, pyruvate oxidation, citric acid cycle, electron transport chain, and oxidative phosphorylation. The only difference is instead of using oxygen as that final electron acceptor, it uses another molecule. So glycolysis still occurs, chemiosmosis still occurs, and all of those steps in between, we just aren't using oxygen. Very often it is nitrogen or sulfur. And so we have different organisms that use different electron acceptors. We have nitrate reducing organisms, we have sulfur reducing organisms. Those are the ones that you kind of smell, that sulfur smell, like by a hot spring or something like that. Those are sulfate reducers. They're using sulfate instead of oxygen for respiration and to produce their ATP. And then we've got methane producers. These are I show those cows down on the bottom. The reason that I show those is they have special bacteria in their guts that produce methane. We all know what methane is and how cows release it. So you can have a little giggle right now if you would like. Overall, it's a little bit less efficient, but in general is still producing a lot of ATP. From this process of anaerobic respiration. So overall summary of cellular respiration. We get 2 ATP from glycolysis, 2 from citric acid cycle. Those are both substrate level phosphorylation. And then depending on the book, even our book has contradicting numbers. You get somewhere in the ballpark of 32, 36 ATP. It really depends on how they're doing their accounting. The discrepancy sort of comes in when you look at FADH2 entering that electron transport chain a little bit later. So if you have more FADH2, you're actually getting a little bit less ATP. If you have more NADH, you're actually getting a little bit more ATP, excuse me, because NADH and FADH2 can actually come from other processes as well. It's not just this glycolysis and citric acid cycle. So there's a range. If you tell me 32, 36 ATP, 38, somewhere in that ballpark for the total, that's totally fine. I do want you to know the general amount of ATP that's produced though, and to understand that the bulk of it is produced via ATP synthase and that electron transport chain in that final step of oxidative phosphorylation. We talk about cellular respiration as this process that produces tons of energy, and it actually does. But it's actually pretty inefficient. So I said that the complete breakdown of glucose yields 686 kilocalories of energy. That's a lot of energy, but most of that is actually lost as heat. So really, we're only getting about 34% of the energy contained in a glucose molecule converted into usable energy in ATP. That might not sound a lot, but that's actually really efficient compared to some other, say, a car engine or something that is converting a carbohydrate or a hydrocarbon into energy. Cellular respiration is actually really efficient, but the rest of that energy, the energy that is lost as heat, and that's what allows animals to heat their bodies. We talked about endotherms and ectotherms in Lab 4. Endotherms can generate enough heat to regulate their body temperature, to maintain a constant body temperature, and that's because of all of the cellular respiration that's going on. Ectotherms still produce energy via cellular respiration. They're just not producing as much, and so they're not releasing as much heat, and so they can't control their body temperature as much. And that's why they need the environment to help them cool or warm them up.